Good morning, everyone. Hi, Etienne.
Hello.
Hi, nice to see you. Hi, Anna.
Hello, good to see you too.
Oh, I know that background. Pretty dark in the office.
Mm-hmm.
Mm-hmm.
I have the cheap spot here on the back, not too close to the window.
Well, ask for a desk lamp.
I got one. Here it is.
Wonderful. Right, we're still waiting for Marsh to connect, who's going to present for us today. And the rest of the team. Did I miss something? Is a holiday in France? Ooh, busted.
It's Monday and very early in the morning.
That's how we do it. Let's see. Here we go. At least our speaker's here. Yes, no, maybe.
Hello.
Hello. Hi, Evie. Is that a cat? Evie? Can you hear us? Yes. Bring your pet to Neuroccino. I don't think we can hear you. Yes, you're muted. Right. Let's make a start. It's 32. Welcome to Neuroccino, everyone. We're live on Zoom and we're live over on YouTube. If you have questions about the paper, you're always welcome to type your question in the chat, and then I ask our speaker for the day, or we discuss it as a group to the best of our knowledge. The rules of the game are that you don't present your own work and you just bring a piece of science that you found exciting, which could be a paper, a book chapter, a book, a YouTube video, any piece of science. Very inclusive here. Today we have someone new present, which is fantastic. Thanks for volunteering, Maj. He is a postdoc with Damien Wasserman in Paris. And I don't even know the paper we're going to hear about today. So if you could share your screen.
Yes, of course.
Go into full screen mode so we can nicely see it. And you can take it away. Just going to make you co-host in case I drop out.
Can you see it?
Yes, perfect.
Okay.
Lovely. Over to you.
So should I present myself first before I starting?
Sure, that'd be nice.
My name is Majd, and I'm a postdoc at INRIA Paris-Saclay, supervised by Damien Wasserman, as Stephanie has said. And I'm here in Bordeaux for first personal reasons, and turned out also for professional reasons. So I'm leading a potential collaboration between Damien and Michelle and the GIN team. And I'm going to stay with you guys for the next year, I hope. And it's nice to participate in my first Neuroccino. And that being said, I hope you forgive me if I do it wrong for the first time. Yeah, so let's get into it. So
sorry to interrupt you. Could you share the link to the paper so everyone can open it if they want to?
Okay. I'm just gonna. All right. I'm going to put it in the chat.
Fantastic. Oh, we actually know one of the authors. He was one of our CNS talk speakers. Thanks.
Okay. So to get into the paper, so this paper came out last year, and I read it earlier this year. I was kind of little bit confused about what paper to present and the level of papers to be presented in this event, but I really like this one. It's about the functional harmonics in the brain and how they bridge together the two popular views of the brain, the modular organization and the gradient organization of the brain. And the motivation behind this paper, can you see me scrolling, right? You can still see the paper, right? Okay, so the motivation behind this paper is that the brain is traditionally known to be organized according to specialized regions that also coordinate and connect to form large-scale networks in the brain. Each network subserves some set of functions, but also recent evidence suggests that the borders between those networks and regions is not sharp. It transitions gradually. And these two views has been in a little bit of a clash in recent years. So in this paper, they proposed that the principle of harmonicity, which is ubiquitous in nature, for example, in light, we can decompose light into different sunlight or white light into different frequencies, as in a prism and as in a rainbow. And those different light frequencies actually are called harmonics. And when we combine those frequencies, we get the light. And in the brain, there are analogously in the brain, there are a set of harmonics, as this paper proposed, that when combined could explain why we have specialized regions and also why there are gradual transitions in the brain between regions. So in the introduction, the authors introduced the topographic organization of the brain as a set of functionally specialized areas, but also present evidence of gradual transitions, as in before in specialized regions such as the visual cortex, there's the retinotopy or the somatomotor or somatosensory cortex, there's the somotopy and tonotopy in the auditory cortex, where representations of the visual field, the body, and the auditory frequency spectrum are spatially continuous. And they also describe that the coherent oscillations or the communication between distinct brain areas give rise to complex cognition. And also not just the coherence, but also the distance from sensory motor regions. So as information converge on association regions in the brain, and as we get farther from the sensory motor regions, we get into more abstract thoughts and abstract representations of our environment. As a consequence, a gradiential organization of the brain might be a general organizational principle throughout the cortex and not just in the primary sensory motor areas. But the principles, those two principles have not been bridged together so far. And what this paper suggests that the human brain's function organization could be governed by the natural principle of harmonic modes. And so we can extract different activation patterns or activity patterns in the brain that each represent a frequency of change in connectivity. And when combined, could also reveal the gradient organization and the network organization. So in this work, the authors uncover 11 harmonic modes. They don't really say why they chose the number 11, but I guess from the amount of variance explained by the different modes across the brain. And so to get into the results and also the methods as well. So how are those 11 harmonics estimated? Basically, the authors compute the dense functional connectivity matrix of 812 subjects from the Human Connectome project, and the dense functional connectivity matrix is describes the connectivity between all the vertices in a surface representation or all the voxels in the brain, not parcels. And the connectivity is measured by Pearson correlation, and in total they have 59,412 vertices. That's why it's called a dense. This matrix is then thresholded such that for each vertex, we only get the 300 nearest neighbors or the 300 vertices that are mostly or strongly connected to it. and it is binarized. So connections that are strong are put to 1 and the others are put to 0. And what do? Is that they compute the graph Laplacian of the connectivity matrix across the 812 subjects. So this is an average matrix over all the subjects. So what is the graph Laplacian? Intuitively, it is it describes the change in the rate of change of connectivity patterns across the brain. So regions differ in their connectivity patterns, and along the whole brain, there is a frequency of change in this connectivity pattern. And the Laplacian of the graph describes how fast does this change happen. And there are different frequencies of changes, and that's what we are extracting here. So these are the function harmonics. They represent the eigenvectors of the graph Laplacian, which is a matrix, as I said. And the first 11 were, here only the first 5 are shown for illustration purposes, but the first 11 were extracted, and they will be shown here. So these 11 harmonics, each one they are ordered by increasing order of frequency, spatial frequency. So what I mean by spatial frequency here, for example, the first harmonic describe changes from the visual cortex to the sensory motor cortex across the whole brain. And the second one describes changes in connectivity patterns from association areas to primary motor and visual areas. So basically, the first 2 harmonics can describe the principal gradients of connectivity that Daniel Margulis have estimated before in his paper. And these are considered low frequency changes in connectivity because they describe changes across the whole brain, not within different regions of the brain. As we go higher in the frequency, we started seeing for example, in the sensory motor cortex here, there's differentiation between regions of the eye, the face, and the hand and the foot. So this pattern goes from this pattern describes the different somotopic representation of the sensory motor cortex. And as we go also to the fourth one, we have the retinotopic representation of the visual cortex. We have the primary visual areas, and then the early visual areas, I mean, and there's a progression across the visual cortex. And as we go to higher frequencies, we also get to the part where we describe where the 11th harmonic describes changes between right hand and left hand. So these are high frequency changes in connectivity patterns across the brain. Any questions so far? Nope.
No, I think it's fine. We do the questions at the end, don't worry.
Okay. So basically what we understand from this figure is that the harmonics can explain both low frequency changes, which are gradients in other terms, and also high frequency changes, which can be described as specialized regions in the brain. So here we find this bridge between the model of functional specialization and the model of gradiential changes in different features of the brain, such as functional connectivity. As we move forward, we can see, for example, if we lay the 11th harmonic and the 3rd harmonic on a graph, on a scatter plot, we can see like a cross shape. So each point represents a vertex in the sensory motor cortex, or in the motor cortex, and we can see a difference between left hand, right hand, foot, and face. So they are on the edges of this cross-shaped layout. And even in the visual cortex, it's the same thing. There are differences between the early areas that show progression from the V1 to V4 using the 4th harmonic and the 8th harmonic as well. So those functional brain harmonics indeed capture many of the aspects of organizational aspects of the brain. Now, how can we know, the authors ask, how can we know that if those harmonics actually reveal specialized regions that are already known to the literature? So what they did is they computed other control bases. So these are called the functional harmonics bases. They computed other eigenvectors from the adjacency matrix, the one that's binarized, the binarized version of the function connectivity, not using the Laplacian. And they also computed the eigenvectors from the original function connectivity matrix. And also they use principal components and independent components. And they computed an index that shows how much are the values homogeneous between different regions of the brain. And the regions were described using the HCP parcellation. And indeed, in the harmonics, so these are the 11 harmonics, the different circles. And indeed, the harmonics show a higher or significantly higher silhouette value. It means that there's more homogeneity within regions, which means that specialized regions are revealed better by harmonics than any other measures of differentiation in functional connectivity in the brain. And another interesting result I found in this paper is that different tasks from the HCP set of tasks could also be reconstructed from the functional harmonics. And so these are the different measures, so functional harmonics and all the other control measures. And functional harmonics actually outperform the others in reconstructing tasks. In this graph, you can see on the y-axis, there's a normalized reconstruction error. And these are the components of the harmonics from all of the different analysis paradigms. And we can see in the red, which are the functional harmonics, as we increase the number of components used to reconstruct the task activation patterns, the reconstruction error decreases. And in red here, the functional harmonics outperform all the others, basically. And of course, as we increase the number of harmonics used to reconstruct, we get much closer to the original task map. So if we only use the first harmonic, it's still not similar at all to the original task map. But as we increase the number of harmonics used from 1 to 100 here in the H panel of the figure, we get much closer to the original task map. And indeed, this further supports this result. So here in A, we have the working memory activation map. So working memory of face versus the average working memory from the HCP task dataset. And if we only use the first or the strongest contributor, so these are the 11 harmonics, and each of them has a contribution value to a task activation map. If we use the strongest contributor only, the task activation map does not correspond a lot to the original one. But as we include more components, we can construct the activation maps. And so I think that's the main ideas of this paper. If we want, we can, if you have questions, we can go deeper to discuss them one by one. What I really like about it is that, for example, in a paper I wrote about the gradients in the lateral prefrontal cortex, I always got reviews about why are using the gradient mapping technique, while it has been known that the brain is divided into parcels, not gradients, and gradients are more of a byproduct of the methods you use. So what I like about this paper is that they kind of say that it's not actually a byproduct. It's only about the different frequency of changes we're talking about. So if we are talking about high frequency changes in connectivity, then yes, we have specialized parcels. But if we are talking about low frequency changes in connectivity patterns, yes, we are talking about gradients. So we are kind of bridging the two ideas together. And thank you.
Lovely. Thank you very much. I love that you already answered my first question, which was why you were particularly excited about the paper, and
Cool.
that very much came across. So thanks for answering that question already. Are there questions in the room, questions over on YouTube? If not, you have to do with my list of questions again.
Sure.
No one? Okay, I'll make a start then. So the fantastic paper, very interesting. Thanks for choosing it. So they used the HCP data set, right?
Yes.
I was wondering if they also validated the method using other data sets, or if there is certain aspects to pay attention to in terms of data sets that could or could not be used for this kind of analysis.
Actually, no, I didn't see that they validated their data on another large data set, although that would be very, very interesting. But they only used resting state data in this case. I think it would be very interesting if we can use, for example, the UK Biobank data to validate this, or even try to validate it on the single subject data using, for example, the Midnight Scan Club data set. But they did not validate it on another data set, no.
That would have been my second question, which is, you've shown this beautiful figure, I forgot which number it was now, where they used the individual, keep going. Sorry for everyone else, this whole scrolling thing is not making it easier. After this one?
You mean the harmonic,
where they used the one, here we go, 1 to
Yeah.
100 reconstructions to
Yeah.
go back to the original task map.
Yeah.
So obviously this was conducted on a group level map, I'm assuming. And I was wondering if there's any appreciation of variability and how easy it would be to reconstruct an individual task map.
Yeah, exactly. Yes. So these are average maps across the whole data set, 812 subjects. Yes, of course, it would be interesting. Well, the way they do it is that they get the activation map first and they decompose it using the same methods as before to the different harmonics, and then they try to use different number of harmonics to reconstruct it back. That I think it would be totally feasible to do it on the subject level. Yes, if we can just decompose the subject level activation maps. Although I'm not really sure about HCP because they don't have a large battery of tasks there, and the duration of the tasks is really short, so we would lose a lot of information from the group level analysis. I think other data sets that have like the deep phenotyping type data sets where subjects do an extensive battery of tasks, much more comprehensive than the HCP one, would be much more interesting to do with this type of analysis.
Great, thank you. I can see that Hervé has a question for you as well.
Thanks so much. It was a really interesting paper. So my question is more it's a naive question. So I'm not familiar with these harmonic, functional harmonic maps. So how will you define these maps? So what I understood is that they represent change of frequency, something like that. So, but biology speaking, how you will define it in a way I can understand it a little bit better.
Okay, so thank you for the question. each region or let's say each voxel in the brain has a connectivity pattern with the rest of the brain.
Yeah.
And it's slightly different from the voxel that's directly beside it. It's not the same. It could be very similar. And as you go away from this voxel, this connectivity pattern changes until you get like to a region that is really far away from it, and it has a completely different connectivity pattern. So there's kind of a gradient of changes of connectivity pattern. But in the harmonics, there are different, let's say, speeds of changes, different frequencies of changes in this gradient. So there are changes across the whole brain. So between large regions, such as the entire motor cortex and the association cortex. So these are called low frequency changes, because they describe changes on the grand level, not on the small or minute level. And as we go further, the changes gets faster. So we have changes within a region. For example, within the motor cortex, we have differentiation. I want to show you this one. Sorry for scrolling again. So in the 11th harmonic, we have changes within the motor cortex, which is differentiates the left and the right hand. So this is a high frequency change of connectivity pattern.
Okay.
So right hand voxels, or in the right region, they are colored blue, and here are colored yellow. So they are mostly different from each other in this frequency band, which only describes changes within a region, which is the motor cortex.
Okay.
But as we go to the first one, the changes are across the whole brain. So in a more grander level of changes. And
in this figure, they ordered the different maps based on these harmonic frequencies.
Yes, but it is also mathematically driven. So these are eigenvectors, and each one has an eigenvalue in the Laplacian matrix of the graph. And so this has the weakest eigenvalue, and this has the strongest eigenvalue, or the greatest eigenvalue and the lowest eigenvalue. And actually this represents the lowest frequency of change to the highest frequency of change. Okay, so I need to digest. Yeah. Tell me that. And a following question. So is it the first paper looking at these harmonic maps, if not, if there is other papers, did they look at clinically speaking at if in different pathologies they found differences, or if they can use these maps to differentiate some substrates of pathologies? It's not the first paper that investigates functional harmonics. There's a study in 2000. They actually cited it. There is a study in 2014 about structural harmonic modes. It was shown that the harmonic modes of the structural connectome, so they get the fusion weighted structural connections, and they do the same analysis, not on functional connectivity. And there have been also the same type of analysis, but within a region, like the visual or visual cortex or the sensory motor cortex, but not on the entire brain. Okay. So this is the first study that uses the dense connectome of the brain, dense functional connectivity of the brain. So between voxels and between the whole voxels of the brain. I don't know about any clinical applications of this, but it would be interesting to go into different populations, clinical populations, doing this analysis to see if there are disruptions that are specific to harmonics, to different harmonics. Yeah. Thanks. Thank you.
Thank you. Final question for me, and then we're sadly out of time. But so you showed that you can reconstruct the original task map, and this seems to work beautifully for primary functions like the visual areas or the motor areas. But I have to say the right-hand label on the left side of the brain is too complicated for a Monday morning to remember which side is which. But is there task maps that are harder to reconstruct? So for example, putting my language hat on, I didn't see any language networks or areas labeled when you scrolled through. So I was just wondering if there's certain task maps that are harder to...
Actually, they kind of reconstructed the language task, I think.
Oh, I missed it.
So the language, yeah, so they
Oh,
used
there
the
it is.
task, but I don't know if they showed any brain maps. I think in the supplementary material, which I don't have.
Okay, I'll dig a bit deeper into the paper then.
Yeah.
Lovely. Thank you so much. That's all the time we have this morning. Thanks for joining us. Thanks for volunteering. We'll be back next Monday morning, 9:30, fresh, bright and early. And if anyone comes across exciting science, just volunteer yourself to present it. And I wish you an exciting week in science. See you next Monday. Bye.
Thank you. Have a good day.
Bye-bye.
Bye.